So this chapter is a very important one. Let's just outline a few things that we're going to be talking here about. So gene expression. Gene expression is where the cell decodes the protein recipe, the protein building codes on the DNA to make proteins. You express the genes, you take them, you follow those instructions, and you make proteins from them. The cell does this. First, we're going to talk about how DNA. We already talked about DNA structure. We'll kind of do a quick review. Then we'll talk about DNA replication. DNA replication, which is DNA making a copy of itself. And it has to do that, you recall, during the S phase of interphase before the cell divides. It has to do that very carefully because the genes don't want to get corrupted as that's happening. Remember, the genes are the protein codes, protein building codes. Then we're going to talk about RNA. Remember, there's messenger RNA, ribosomal RNA, and transfer RNA. We didn't really go into it all yet. So there's different structures and functions of those for transcription. Okay, well, transcription is taking the DNA gene and turning it into a copy, just that part of the DNA, but in RNA form. So taking the DNA gene and turning it into a messenger RNA, this happens in the nucleus. It will leave the nucleus, and that's the next step. Okay, once it leaves the nucleus, it will be translated. Translation is taking the mRNA instructions, which are a copy of the DNA gene, and transforming it into the protein that the message has the instructions to build. Okay, this requires ribosomal RNA and tRNA as well to help decode the instructions on mRNA. Then we're going to talk about mutations. Mutations are mistakes that happen during this gene expression process, or maybe it happens during replication. So mutations are permanent changes in the DNA. Basically by accident. This happens randomly at a low frequency, but it's important that it happens. We'll talk about that when we get to evolution. Okay, so let's hit gene expression. Gene expression is when DNA, basically the whole process of DNA codes, called the genes, the instructions for making specific proteins. When that code gets decoded and the protein is formed from those instructions, that is gene expression. So, in brief, when DNA is used to make specific proteins. this is a this this string of beads right here in the picture is a polypeptide, a protein. And each of the monomers are amino acids. Now there's 20 different amino acids. So that's like 20 letters in the protein alphabet. So in the protein language. So the order of those amino acids is coded for in the specific gene on the DNA molecule. The DNA molecule in that gene has the order that the amino acids should be and what amino acids they are and how long the polypeptide should be. So all that information is coded for in the gene, in the DNA. So gene expression is a two-step process, generally speaking, where first DNA turns into RNA, specifically mRNA. mRNA carries the gene code to the cytoplasm. And there in the cytoplasm, the mRNA works with the ribosomal RNA and the tRNA, transfer RNA, to form a protein from those instructions that mRNA copied from DNA. So transcription is the copying of the DNA gene into mRNA. Transcription. And then this process right here is called translation. Both are steps of gene expression. Translation, again, is decoding the mRNA into the protein. Okay, so here it's written again. Now, DNA has this little curved arrow showing that that's representing DNA replication, which is not part of gene expression, but it's important that DNA copies itself, as you found out in the S phase of interphase of the cell cycle. Okay, so this gene expression outline right here is called the central dogma. It is central because it applies to any living cell, bacterial, human, fungi, plant, doesn't matter what the organism is, it follows this central dogma. Dogma is an established fact. So the central dogma states that this is the direction of gene expression. DNA information to mRNA information to proteins. You don't go the other direction. Proteins don't change the mRNA. mRNA doesn't go back and do anything to the DNA. Okay, so both transcription and translation, we're going to use these stages for both of them. Initiation means the beginning of the process. Elongation is whatever's being made lengthens. The molecule becomes a bigger and bigger polymer. Okay, so in transcription, it's RNA. It's the mRNA that is being elongated. In translation, it's the protein that's being elongated. And then we have a stopping point called the termination, where the chain is completed. Okay, all this is saying is in prokaryotes, you don't have a nucleus. So it all happens in the cytoplasm, gene expression. All the steps happen in the cytoplasm. In eukaryotes with a nucleus, the DNA is protected by the nuclear membrane, because that's what's inside the nucleus, the DNA. And inside the nucleus, what happens is where transcription occurs. Making the mRNA from the DNA gene to mRNA. And then outside in the cytoplasm, this is for eukaryotic cells, in the cytoplasm, actually on the ribosome, which is either attached to the rough ER or free-floating in the cytosol. In the cytoplasm, what happens is translation, where the mRNA message is turned into protein. Okay, but with the help, of course, of ribosomal RNA and transfer RNA. Okay, so this big blue thing right here is the nucleus. So this is just showing that transcription occurs in the nucleus if you have one. Then translation occurs in the cytoplasm. Okay, so there's a little step here that I didn't tell you about till now. So once the RNA ribbon is made, transcribed from the DNA gene, it's called pre-mRNA. And then its pieces are cut out of it, and then the remaining parts are glued back together, or bonded back together, until you have the finished mRNA product. So it's processed. It's processed after it's been transcribed. Once it's processed, it's the actual message edited properly. It comes out of the nuclear membrane, attaches to a ribosome, either free floating in the cytoplasm or stuck on the rough ER. Once the mRNA gets on the ribosome, with the help of tRNA, the protein, polypeptide, remember, is a protein, is built. Now, how does a cell know what proteins to make? No, this is all controlled by chemical signals, maybe hormones tell a cell to get moving and make this particular protein. Genes can be turned on if the proteins, the histones that connect to the protein, are released around the specific gene that needs to be copied, transcribed. There's all kinds of ways to regulate transcription and translation, but we're not going to get into that detail. Just know that a cell is not crazily making every protein nonstop from every gene in the DNA. No, it only makes what it needs at the time. Okay, so let's start with a very simple regulation of gene expression in bacteria. Okay, so bacteria, a certain kind of bacteria will eat lactose, the lactose in milk as milk sugar. And that's what makes milk sour eventually. The bacteria inside the milk that you really can't get 100% rid of will eventually make the milk go sour. So, lactose in the milk. If there's plenty of lactose present, the bacteria wants to make the genes for the enzyme that breaks down the lactose and turns it into glucose. Okay, so but when lactose is absent, it's a waste of energy to make that enzyme, so those genes are turned off for making the enzyme that breaks down the lactose sugar. So look at this picture here. This number 1 is RNA polymerase. Okay, that's RNA polymerase. That is the enzyme that will transcribe the genes for lactose breakdown, for the enzyme lactase, which is a protein coded for by these parts of the gene, that the lactase gene. Lactase is the enzyme that breaks down lactose. Okay, so this is the DNA. Those are basically the genes, the parts of the genes for the enzyme that will break down lactose sugar. So number 2, number 4, let's look. Number 3 is the promoter. You can look at my code down here. Okay, the promoter is where the RNA polymerase lands on and starts transcription. Starts making mRNA for the lactase enzyme, the protein that will break down the lactose sugar. Okay, look at down here at the index. Number 4 is the operator. It's on the DNA. It's part of the DNA, but it is a specific part at the beginning. It's of the lactase gene. Lactase is the enzyme again that will break down lactose. But this part right here is a regulator gene. It's called an operator. The operator right here has a part that fits into this green block, which is the repressor protein. Okay, that's another protein and it fits the operator. So number 2, the repressor fits the operator. And when lactose is present, lactose are these little crosses. That's the sugar that needs to be broken down. When that's present, it will attach to the repressor protein and disconnect the repressor from the operator. Okay, so usually the repressor protein is sitting there blocking the way of the gene, turning off the gene for lactase, the enzyme that breaks down lactose. Why should you make lactase if there's no lactose to break down? So the repressor protein sits there on the operator Okay, now, if there's a lot of lactose present, as you can see here, these crosses, then that lactose will connect in there, much like a substrate does an enzyme. And it will come off. The whole thing will fly off. It will, and then the operator will be open and will allow the RNA polymerase to go down the DNA molecule and transcribe that gene. Okay, you might say, well, where is the lactase enzyme? Well, that's a protein. You're not going to make that until this mRNA that the yellow blob is causing to form as it transcribes the DNA, which they don't show the messenger RNA ribbon at all because it just started. You don't see the lactase either because that ribbon mRNA has to go it has to go into the cytoplasm and get connected to a protein first. Okay, that's kind of complicated, but we should be aware that is an example of regulation on the bacterial level. That is regulation of transcription. Okay, when we get into eukaryotic cells, the gene library, the number of genes in the nucleus is much, much greater than the gene library in the single chromosome of a bacteria cell. So get used to this library of all the genes. That's called the genome, the organism's genome. All the genes on all the chromosomes. Whether you only have one single chromosome, like bacteria cell, or have 46 chromosomes like we do. Of course, in our more complicated DNA, we need much more regulation than bacteria cells. So the regulators are much more sophisticated and varied because you only want a few genes turned on at a time. You don't want to be wasting energy making all these proteins that you don't need. And of course, different types of tissues in our body make different kinds of proteins. Like a skin cell makes totally different proteins than a gland cell that makes and secretes hormones, than a muscle cell. This is specialized cells have a ton of their other genes turned off. And we'll never use those genes. Okay, every cell in our body has the same chromosomes with all the genes you would need, but most of the genes are turned off, especially if you're specialized. You're differentiated for special tasks like muscle or nerve. Okay, again, muscle cells are contractile. When they're signaled by a neuron to contract, they will. And then nerve cells, like brain cells, look how different they look, and their function is totally different. So they send nerve signals to other cells to do this or that, to communicate, to contract, to start making this hormone, adrenaline, for example. So the point is that no matter what kind of cell we have in our body, each cell contains the same type of DNA. The whole genome is in every cell, but only some of those genes are on. Most of them are turned off. And if you're a given cell, you're never going to turn on certain genes because you'll never make that. Your job is not to make that protein. So this, for eukaryotes, this is just a few examples of ways regulation, gene expression is regulated. So some of the chromosome will stay packed up with the histone proteins. And if you're tightly packed with your proteins, your DNA is not exposed to RNA polymerase, and you cannot make mRNA. You're all locked up. So some genes are just turned off that way. The proteins that loosen up on chromatin and make it soupy are still connected to the genes that you don't need to be on. That's one way. Transcription controls, there's factors, just like there was the repressor protein, more sophisticated, but transcription factors that are proteins that guide the RNA polymerase to and open up the DNA, make the gene accessible to the RNA polymerase, which is the enzyme that does transcription. And then when the mRNA is processed, after it's transcribed, it can be cut up. It can be modified by other enzymes to make it work more efficiently, or to make it not work. So even after the mRNA is made, you can tweak it by mRNA processing, to turn it off or to make it useful. Gene expression is transcription and translation together. So making the protein or RNA coded in the nucleic acid. So that is translation, making the protein, and making the RNA coded in the nucleic acid, that's transcription. So that has to be right. Making amino acids, they don't make amino acids, they pop the amino acids together. Making tRNA, well, that's transcription, making tRNA, which we haven't really talked about. It's not just mRNA that's made, but it's mRNA that translates into protein. But tRNAs also need to be made in the nucleus by transcription, because you'll find out that it's important for translation of proteins. Okay, folding of the protein. No, you don't. That does it on its own because of the R groups and the polar parts of the molecule. So letter A. Okay, the central dogma is DNA is transcribed to mRNA, and the mRNA is translated to protein. That's central dogma. So briefly, right here, letter B. Gene expression is different in prokaryotes and eukaryotes. Okay, ribosomes are different in the prokaryotic cell. I didn't say anything about that. No. Prokaryotes do not have a nucleus, of course, so transcription and translation can occur simultaneously in the cytosol or cytoplasm. That is totally true. Eukaryotes carry out translation in the nucleus. No. They do transcription in the nucleus. Transcription and translation in the cytoplasm. So that's totally wrong. prokaryotes have more than one chromosome. No. One circular chromosome. No two cells in one human body have exactly the same DNA. That is totally false. Now, DNA in one cell has different genes turned off and different genes turned on than the DNA in another cell, but every cell has exactly the same DNA. The first control point in eukaryotic gene expression, well, that would be when transcription is occurring. That would be the transcription factors, which we talked very briefly about. Where does transcription occur in eukaryotic cells? Inside the nucleus. Once the DNA is unwound from the chromosomes, that means the histone proteins have come off. Which of the following is necessary to begin transcription? Transcription factors and RNA polymerase. Okay, that's for eukaryotic cells. Once transcribed, copied from the DNA, eukaryotic mRNA typically undergoes substantial alternation. That's called pre-RNA processing, where pieces of the RNA segment, the mRNA segment, are removed. Okay, the rest is like bleh, whatever. Ribosomes, no. Transcription doesn't occur in the cytoplasm order. Ribosomes are linkage to histone. No, the histone doesn't connect to mRNA. It connects to DNA to turn off genes. Fusion into circular forms, no. Plasmids are little rings of DNA in bacteria. So we're talking about eukaryotes. So letter A. Okay, so we already talked about DNA structure. I'll just review that quickly and add a few points I didn't say before. We're going to talk about how DNA is replicated, so we're going to leave transcription for now. Okay, DNA was the structure of DNA. Once you know the structure, you know how it explains how it functions. So the structure was made 3 years before I was born. The structure was discovered by Watson and Crick. Crick and Watson. Okay, you should know that. Blah, blah, blah. We'll talk about that. But it was really Rosalind Franklin who discovered the double helix, who gave chemical evidence for DNA's double helix structure. These two guys stole her evidence. They kind of, it's a bad story. They took her evidence, didn't give her credit for it, and they used it to explain how DNA replicates. Okay, DNAs, this is the monomer of nucleic acids, and DNA is a nucleic acid, so is RNA. So, remember, let's just review this. Nucleotide has a phosphate group connected to a sugar, a 5-carbon sugar. 1, 2, 3, 4, 5 carbon sugar. Remember, they don't put numbers. They don't put the carbon ends even because it looks messy. Okay, that sugar on the other side is connected to a nitrogen base. I call it nitrogen base. And that could be A, T, C, or G. Adenine, thymine, cytosine, or guanine. DNA looks like a ladder. This is a twisted ladder, a double helix, but I untwisted it here. So we got the sugar phosphate backbones as the rails. I call them rails. Here's the other rail. Okay, and the rungs of the ladder are the nitrogen bases. The nitrogen bases make up the rungs, the colorful stepping places, the rungs on the ladder. Okay, so distinguish what parts of the nucleotide make up the rail and what part makes up the rungs. The rungs of the ladder. Okay, the nitrogen bases are held together by hydrogen bonds, weak bonds, but when there's a lot of them, it holds them together, like we talked about before. The sugar is deoxyribose. That's where we get DNA. Deoxyribose. Okay, the 4 types of bases we talked about. Now, let's go on. So there's the backbones, rails. It's a twisting ladder, a double helix. And the rungs are the nitrogen bases. And you should know that the rails are sugar phosphate, or sugar phosphate groups. Okay, this is a simple picture here. Now, here, sugar phosphate backbone or rail. The nitrogenous bases make up the rungs. Now here's the base pairing rules. Just remind you, adenine has to always go with thymine, and cytosine always has to go with guanine. CGAT at GC. So know the base pairing rule right here. At least that's the rule for DNA. Okay, these bases, when in the rungs, are complementary. Complementary bases basically just fulfill the base pairing rules. So 2 bases are complementary if they will combine together to form a rung in the ladder. In other words, if they follow the base pairing rules, you will not see T connected to G anywhere, etc. Okay, what does anti-parallel mean? DNA is anti-parallel. Well, parallel means these 2 rails are parallel to each other, even if it's twisted. Okay, but look how one's upside down. If you think of the house right here, if you think of the deoxyribose sugar, it looks like this. There's a carbon right there too. 1, 2, 3, 4, 5 carbons. Well, that's oxygen at the roof, at the peak of the roof. So if we go around, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Now, carbon 5 is easy because he's connected to a phosphate group. Carbon 3 is the other side, the bottom, right below the phosphate group. So carbon 3 is also important, Well, they're all important, but we're picking these two numbers. So carbon 5 and carbon 3. So carbon 5 is that one next to the PO4 group. And carbon 3 is like a sidewalk leading up. Think of that. That would be the chimney of the house. That would be the sidewalk leading up to the bottom of the house. Okay, so what antiparallel means is if you have the 5' end sticking up right here, connect to the phosphate group, you'll have, at the very bottom of the molecule, you'll have the 3' end sticking out. So if the top end is 5', the bottom end is 3'. Now look, the way this is connected, look across like this. The way it's connected, there's the oxygen group. There's the, let's see, 1, 2, 3. There's the 5' under the phosphate group. There's 5' N. And there's the 3' end right there, like the sidewalk leading up to the house. Okay, this would be like the chimney. The 5' would be like the chimney. So think of the house like this. Okay, there's the phosphate group. 5' and the chimney. Think of that as the chimney on the house. This would be like the sidewalk. Okay, so don't want to complicate this too much. You could, instead of saying the chimney, you could think, well, that's the one that's connected to the phosphate group. That's connected to the phosphate group. If you want to do it that way. So that's 5' carbon right there. 1, 2, 3, 4, 5. Remember, the carbon's sticking out of the ring. Okay, don't want to make it complicated. Okay, so there's the 5' phosphate 5, phosphate 5. Think of it that way. They sound the same. Phosphate 5, phosphate 5. Okay, so here's the 5' end. And here's the 5' end, but it's upside down, so this would be the 3' end. So those are opposites. That's what they mean by antiparallel. The one rail is flipped around from the other rail. Basically, that's all I'm saying. Over here, you can see that this would be 5', so the 3' is sticking out right there. The 3' carbon's right there. Oops. Right where I showed it. 1, 2, 3. Okay, so that end is the 3' end. The sidewalk is the 3' end, and that's what's sticking out. And right here, we got the 5' end because it's connected to the sugar. So notice the 3' end and 5' end are opposites. Actually, it's this one right here. These are base pair. This would be the 5' end. So would that, but let's keep it so we don't have any leftover part. Okay, so whenever you have a DNA molecule, let's say it's a DNA molecule. If this is 3', this would be 5', this would be 3', this would be 5'. You should be able to understand that. So once again, the 5' carbon is connected to the phosphate group. I think of that as a chimney. The 3' is carbon is like the sidewalk leading up to the house. That carbon right there. Okay, so let's talk about DNA replication. Okay, so when a cell's getting ready to divide, they must replicate their DNA during the S phase of interphase. Okay, I hope you remember that. Okay, so the first stage of DNA replication is to unzip the two DNA strands. And that's kind of easy because hydrogen bonds is what's connecting the two DNA strands. So break the hydrogen bonds and unzip it. That's what helicase, the enzyme helicase does. It's like your finger going down and making the double-stranded DNA into single-stranded DNA. Each strand serves as a template for creating a complementary strand of bases. Okay, so this can be copied, transcribed, as can this. Okay, so that's how DNA is going to get transcribed. First it's unzipped, then each rail with its nucleotides attached will serve as a template, or what's a template? A template is something that you use to copy something. So we'll show that in the next slide. Okay, so next step is for DNA polymerase to enter on. Okay, so here's the original strand, the template, like I just talked. And here's the new strand forming. Now to do that, you have to have a big enzyme called DNA polymerase that reads the codes. It reads the nucleotides. So let me make that bigger. It reads those nucleotides on the template and fits in just the right one. Puts the right, this is phosphate group, ribose, and nucleic dimension base. And it fits it on according to the base pairing rules. So that enzyme is called DNA polymerase. I kind of talked about RNA polymerase before when I was talking about transcription. We'll get back to that when we get to transcription details. But right here we're talking about DNA production being copied from DNA. That's not transcription. That is replication. Okay, this is replication. So, what we see here, they're not showing the DNA polymerase, but it's there on both ends. So, let's draw it. There's the DNA polymerase on both ends. The DNA polymerase reads in the 3 to 5 prime direction. So let me just write that. It reads the DNA molecule, the template, 3 prime to 5 prime. But it makes new DNA because the new strand is, what do we say, anti-parallel. So it makes DNA, the new DNA, in the 5 prime to 3 prime direction. So it reads the DNA polymerase, DNA polymerase means an enzyme that makes a polymer. DNA polymerase reads the template DNA in the 3 to 5 prime direction, but DNA polymerase makes the new DNA strand in the 5 to 3 prime direction. Now, this is important, a term. Each new DNA has one strand from the original molecule, the template strand, and then the other strand is a new strand. So this is called semi-conservative replication. Semi means partially saved. So what happens is, like I just said, one strand is the template strand. And let's say, I don't know, 3 to 5, there's a template strand with all its nucleic acid, nitrogen bases matching the new strand. Okay, this is the new DNA strand. And he is complementary to the template strand. And by 5 prime to 3 prime. Which way is it made? This direction, right? Or which way is it read? That direction, 3 to 5. The template strands read 3 to 5. But the new DNA is made 5 to 3 prime direction. What is the shape of DNA called? In DNA A, okay, A, T, Z, G. I just remember that. So A pairs with T. DNA guanine, okay, A, T, C, G. So guanine pairs with cytosine. If one strand is C, G, G, T, A, C, those are the base nitrogen bases. The complementary strand, following the base rules, would be G, C, C, A, T, G. So I just look at the end or the beginning. So choice D. One strand DNA is HE. The complementary strand would be follow the base pairs. T, C, G, A, C, T. So I picked the one that ends with ACT. Okay, the parental strand, that's the template strand, is 5 prime, blah, blah, blah. Okay, so they're putting these numbers in, so what would the daughter strand be? Well, if it's 5 prime here, it's 3 prime for the new strand. The daughter strand is the new strand. Okay, so that'd be T, A, G, and let's see if anything starts with that. So we got 2 choices. Keep on. I do the last one. No, let's do the next one. C, T, A. So, okay, it has to be that guy. Oh, see, that's why I like to do the last one. T, A, T, G. So, ATG. Oh, that one works too. Oh, I see. These are the same. These are the same. So it has to be one of those two, but because it's anti-parallel, it has to start with 3 prime. So it's this one that's the right one because these are backwards. Okay, let's remember that RNA, there's 3 types. There's messenger RNA, the one I've been primarily talking about, ribosomal RNA, the RNA in ribosomes, and tRNA, transfer RNA. We'll be talking about each of these guys in this part of the unit. And remember that transcription is when DNA, a gene, not the whole DNA molecule, a DNA gene is copied in the language of RNA, messenger RNA. Okay, RNA is a nucleic acid like DNA, so it's composed of nucleotides. And the nucleotides, again, are, this looks like the sugar, the phosphate group, and then the nitrogen base, and that's the nucleotide, the monomer of a nucleic acid. Now, so how's RNA different? Well, that sugar is ribose. That's why it's called RNA. Whereas the sugar in DNA is deoxyribose. Okay, RNA is a single strand of nucleotides or a chain. Remember, DNA is a double strand. And there's all kinds of shapes for RNA, depending on what type it is. So messenger RNA is just single-stranded. Ribosomal RNA, the RNA in ribosomes, is globular. So it's like a tangled thread ball or a hair ball. And then tRNA has parts that are double-stranded and parts that are single-stranded, so it looks like a T. So it has parts that are like double-stranded. Okay, those are the hydrogen bonds between nitrogen bases. Then parts that are not, that are single-stranded. So I call that T-shape because that's what it looks like. So clover leaf shape or T-shaped. Now, another important difference is instead of thymine, the nitrogen-based thymine has uracil replacing it. So the base pairing rules would be CG still, because RNA still has C and G in it, but there's no T. So adenine will bind with instead of T, because there is no T in the RNA. So where the RNA is double-stranded, if you had A on one of the strands, it would it would base pair with uracil, the nitrogen base uracil, not T. There's no T in RNA. Okay, so there's the base pairing rules for RNA. So this is the same as for DNA, but again, there's no T to match the A, to base pair with A. So you would instead. Okay, so transcription, once again, is taking a gene on the DNA and rewriting it in RNA language. So primarily we're concerned with messenger RNA, but tRNA has to be transcribed from genes on DNA, as does rRNA. Okay, I'm going to skip the whole discussion of how transcription knows to start on the DNA molecule at a particular place. That's transcription factors, are proteins that are produced and go into the nucleus and guide the enzyme that makes the RNA as it copies it from the DNA. It guides it to the right gene. So I'm going to skip that step, initiation. Okay, so on DNA, there's where the gene is on the DNA. That part of the DNA is a template strand. Okay, in replication, there's two template strands, but in transcription, when you make an mRNA, there's only one template strand. The other one's ignored. So, RNA polymerase is that enzyme that will read the template strand and then it will make, it will fit in RNA nucleotides that match the DNA nucleotides. This is kind of confusing. The strand that's not being copied, the DNA strand of that gene that's being copied is called the template strand, but the other strand that's not being copied, the other strand of DNA is called the coding strand. That doesn't make sense because the template strand is what's being decoded. Well, why they do that is because the letters on the RNA, messenger RNA, match the letters exactly on the DNA, except that there'll be a U instead of the T. I'll show you this in an example. So here's the DNA strand. Let's say this is the part of the gene that's being transcribed. Okay, so if that's the template strand on top, it reads 3 to 5. So RNA polymerase comes in here. We make it like this. RNA polymerase comes in here and reads the template strand. Let's say that's RNA polymerase. So it's going to read 3 to 5. And let's say it decides that the template strand is this one right here on top. So it's going to fit in, starting 3 to 5. It will place nucleotides, but not a T. It will place RNA nucleotides, which are the same as DNA, except for uracil instead. That replaces thymine. So this would be G, A, G, G, U, C, A, T. Now, you can see that it matches the coding strand. Okay, T, T, A, A, C, C, but it doesn't match where U's are, because DNA doesn't have a U. Okay, so everywhere else it matches. That's why it's called the coding strand, even though it is not the one that's being read. It is similar, very similar to the mRNA. I should have said that. This is the that red letter stuff is the messenger RNA molecule being made, transcribed from the DNA template strand. Okay, so this would be 5 prime. I keep on doing that. This would be 5 prime. This would be the 3 prime end. Now, that strand does not, that's the mRNA. Newly made mRNA transcript is called. Okay, now that's not going to stay bonded because the RNA polymerase, as soon as it builds it, it feeds it out of itself, like a little snake coming out of them. So it doesn't remain stuck to the DNA. So you recall that the DNA molecule is a twisted ladder, a double helix. So those bases are protected in there. So how does RNA polymerase get in there? Well, I already told you the initiation factors, transcription factors get in there to a specific place and unzip the DNA and make a little bubble, as I think the next picture will show. So here we see the transcription factors, which are proteins, guiding the RNA polymerase to the promoter region, which is the beginning of the gene, the landing pad for the RNA polymerase. Okay, so it's guided there. And RNA polymerase will insert itself and start unzipping the DNA molecule as it reads 3 to 5. So if it's reading 3 to 5 and it's landing right there, this would be the template strand, because that's the one he reads. That's the direction that transcription goes. 3 to 5. I better write that. Okay, so the DNA is read 3 to 5, but RNA is made, the messenger RNA is made 5 to 3, which you'll see, like in my previous picture, you'll see it coming up. So we're going to get a bigger picture of that. Before we continue, let's just compare the DNA strand, the non-template versus the template. Does it contain a gene? Okay, the template strand is what's read by the mRNA, so that's where the gene is. The non-template strand is called the coding strand, and no, it does not contain this gene. It's not what's read. Does RNA polymerase attach to it? Yeah, the template strand it does, but no, it doesn't attach to the non-template. Does the non-template get transcribed in mRNA? No, but yes, the template strand does get rewritten as RNA. So here we see RNA polymerase reading the template strand, reading in the 3 to 5 direction, and making new RNA, the blue strand, in the 5 to 3 direction. But it's reading the template strand in the 3 to 5 direction. Okay, so That's kind of tedious to learn. Now, as RNA moves in the 3 to 5 direction of the template strand, it unzips the DNA. But also it spits out the new RNA as it moves. These are hydrogen bonds between the nitrogen bases, so those are weak bonds. So as it moves, it just pulls it off there, pulls it off the DNA molecule. It remains connected where the RNA is doing its RNA polymerase is doing its work. So when it hits the termination sequence, there's a certain sequence of nucleotides on the DNA that tells it to stop. And so the RNA polymerase will fall out, will come off the track, the DNA will snap back together, and the RNA, the messenger RNA, is released. Just like I said before, this is just like in DNA replication. RNA is made from the 5' end of the new RNA to the 3' end, but it's read. It reads the DNA in the 3 to 5 direction, but it makes new RNA in the 5 to 3 direction. So it reads the template strand in the 3 to 5 direction, but it makes RNA in the 5 to 3 direction. So you should know that little annoying detail. Okay, let's step back and just compare replication with transcription. DNA replication. 2 new double-stranded DNA molecules are produced. Well, half new, half old, I should say. But you get one ladder here. What they're trying to say is you get one ladder. Here's the DNA before replication. And then after replication, you get two molecules of DNA exactly alike, except it doesn't really matter how I'm drawing it, except half of it's old and half of it's brand new. We call that what? Remember? We call that semi-conservative replication. Okay, but in mRNA, you only have one single strand of DNA produced. Okay, we already know this, base pairing rules. A bonds with T for DNA when DNA is copying itself, replicating, but uracil replaces thymine in the new RNA. You don't have T's, you have U binding with the A instead of T binding with the A. Okay, also, the entire DNA molecule is replicated in DNA replication, but only a small piece with the gene of the DNA is transcribed. Only the DNA strand, only a small bit of the DNA strand with the gene, the part with the gene is transcribed. But I should say, synthesis, the formation of the new strand, whether it's DNA or RNA, is always in the 5 to 3 direction. Okay, RNA is not more stable than DNA. DNA is protected in the nucleus, okay? Not only is it protected in the nucleus from enzymes that can digest it, it has a double helix shape, so that protects the bases from getting disrupted. Also, I didn't say this, but T, the nitrogen-based thymine, is more stable than uracil. So if you have T, you're not gonna have mistakes as the T breaks down. Like uracil is more apt to chemically react with things. So also, you don't want DNA to get, you don't want the original documents, the original protein recipes to get damaged, because they're the real thing. The messenger RNA is out in the cytoplasm. It needs to get digested by enzymes eventually, so that we don't keep on making protein off of it. So it's important that RNA is less stable than DNA. It needs to be broken down when enough protein has been formed. The transfer of genetic material from DNA to RNA is transcription. The bases that are read by RNA polymerase are located on the template strand. Okay, what is function of the promoter sequence? Okay, promoter sequence promotes transcription. It's the landing pad for the RNA polymerase, so it can start transcribing the gene into RNA. Okay, the strand that is not transcribed into RNA is called the non-template strand, also called the coding strand, which I don't like. Okay? Okay, sorry, but I did this without the microphone on. So let me try that again. So they give you this gene right here. Now, what is the RNA strand produced by transcription? Well, first of all, be sure you make it anti-parallel. Put a 3' above the 5' or below it. Now, if this is the DNA nucleotides, remember DNA has A, T, C, G. What would be the RNA ones that match? Well, RNA doesn't have T, so what would match with A would be U. What would match with T, still A. What would match with C, G. What would match with the DNA G would be the RNA-C. So just go down the line. A, U, T, A, U, G. G on the DNA would be C, A, U, T, A, etc. So let's look. 3' UAU. 3' UAU. No one else has that. This is supposed to be 3', so it's letter C. Would be the mRNA transcript. Okay, this one I forgot to push the microphone again. So put the 3 above the 5 or below, it doesn't matter. Put the 5 on the other side of 3', and then just start checking out. You can't have T's in RNA, so those are U's. So you look for something 3' UUU, 3' UUU. I don't see it anywhere else, so to save time, I just pick letter C. Okay, if the non-template strand of DNA is this, what is RNA strand produced through transcription? So what I had to do is first do the DNA. Okay, I mean, that's how I do it. So here's the DNA. In the previous problems, I was doing mRNA. So we said this is the non-template strand. Well, then this would be, up here would be the template strand. The one I'm starting here. And I have to, since it's DNA, I have to use DNA nucleotide. So I'm not going to put a U there because it's DNA. T, C, G, C, T, A. Not a U, T, G, A. Okay, so that's DNA. Now I'm looking for what's the mRNA strand. Okay, why did I do that extra step? Why didn't I just do RNA off of this? Because that's a non-template. It's RNA polymerase reads the template. So I had to do my little base pairing with the template strand. So that'd be 3', 5', A, C, G, A. Can't put a T there, so it's U, A, C, T. So ACG, 3', ACG, and then keep on going just to check AUU. Okay, UAC. Oh, I made a mistake. I put a T. Okay, so it has to be this one. Now, I just do the first 3, and I double check, and I check. But be sure you get the prime number correct. Translation, just to review, is putting the messenger RNA information into a protein language. So that's where they get the word translation. Going from nucleic acid to protein, you're translating the message into another language, proteins. So remember, in transcription, DNA language was put into RNA language. Then in translation, like I just said, RNA language is put into protein language. So there's 4 letters that the DNA language can use. A, T, C, G, right? There's 4 letters that the RNA language can use. A, U, C, G. And then proteins have 20 letters, those being amino acids. It takes 3 letters in mRNA to code for 1 amino acid. So 3 nitrogen bases consecutive on the mRNA will code for one amino acid, depending on what the three letters are. The three nitrogen bases depends on what amino acid is going to be placed in the protein, the new protein. So we call these three letters codons. These three nitrogen bases, we call them codons. So the codons again code for one amino acid in the protein. Now there's start codons. Again, codons are on the mRNA. So it would be the nucleotides. Here's a codon, here's a codon, here's a codon, the next codon, etc. Again, that's mRNA. Some codons don't code for an amino acid. Some codons just signal the start of protein synthesis, and some codons code for the stop or the termination of protein synthesis. Okay, so there's 4 letters. There's 4 messenger RNA letters, and there's 3 nucleotides per codon. So statistically, there should be 4 times 4 times 4, 4 to the 3rd power, or 64 different codon combinations with the 3 letters. But there's only 20 amino acids. So there's going to be some codons that code for the same amino acid, because look, you have 64 possibilities, but you only need 20 plus a stop one, a stop codon, and a start codon. So you really only need 22, but there's 64. There's redundancy in the code. Redundancy means repetition. In the genetic code. Okay, so the universal genetic code shows what codons code for what amino acid. So this stuff right here are the amino acids. And then you'll have some stop and some start codons as well. Like here's stop. Okay, so the start codon's not obvious, but the start codon codes for methionine. Methionine is always the first amino acid to be placed in a protein. So it's easy to remember because August is when we start school, right? So you'll never forget that. So start codons, UG, all the other guys. Okay, we see the stop ones. Besides those 4, the rest code for amino acids, one of the 20 amino acids. Okay, they're saying it's universal. This code is used by every life form. They also put viruses, but viruses cheat. They're not living. They just use the equipment in the cell, the host cell that they've invaded. So, but they use it. So, but they're not. Remember, viruses aren't living. So this is strong evidence. This is a unifying factor of all the different types of life forms that have existed and exist now, that they share this same genetic code, is support that all living things share a common ancestor. We'll talk about that in evolution. What is a codon? Well, it's a 3-base sequence on messenger RNA. Okay, so for UAA, the first letter means it's somewhere in this row. The second letter, A, narrows it down to somewhere in this row. And the third letter, A, points this one out right here. Okay, so that's a stop codon. Okay, the same thing. G, it has to be somewhere over here. And then narrow it down with G again. It has to be somewhere over here. And then narrow it down even more. G, it has to be this one. So all those are glycine, GLY. Okay, the codon GAC, so find the first letter G. It has to be in this row. A has to be in this column, particularly right here. Has to be one of those. And then the last letter is C. So I just follow along. And there it is. It has to be that one, GAC. So it's aspartame or aspartic acid, ASP. Okay, MET. You could use the genetic code and see that the codon for MET is AUG, and that you should know is a start codon. So it's letter B. Okay, once again, translation is when RNA is used to build proteins. Polypeptides are proteins. Basically, they're discriminating here that polypeptides are less than 50 amino acids, proteins are more than 50 amino acids, but we'll use those two terms interchangeably. I'm not going to be picky about it. Okay, here's the 3 types of RNA. We already talked in detail about messenger RNA. Let's talk about, again, each one. Okay, so mRNA is single-stranded. It is made like all the RNA is transcribed in the nucleus from genes on the DNA molecule. It's then processed in the nucleus. Maybe little pieces are cut out and then it's spliced back together, the remaining pieces. And then it exits the nucleus into the cytoplasm, where it attaches to a ribosome. Okay, so the ribosomal RNA is globular in shape, right? And it's connected to all kinds of proteins, ribosome proteins. Okay, so it's a dense little ball-like thing, either free floating around in the cytosol or attached to the rough ER. Now there's 2 subunits, a small subunit and a large subunit. Okay, see this little groove right here? That's where the mRNA is gonna be fed through. This is like a train track, and mRNA travels through that little groove. these two subunits are not connected until the mRNA attaches. Then when the protein is done, if there's not any other mRNA, these two pieces, these two subunits of the ribosome separate, releasing the messenger RNA and the protein polypeptide. Transfer RNA. Think of a truck. Transfer. It carries particular amino acids to the ribosome, which will then, the tRNA will then bind those amino acids together with peptide bonding. So, like I said before, tRNA is single-stranded, but there's parts where there's hydrogen bonding to make double strands to give it this clover shape or T shape. So that helps you remember what it looks like. Now, there's two really important parts to this. There's the anti-codon. Anti, not codon. So think of the T as the T in tRNA. So anti-codon is complementary. It base pairs with the codon on the messenger RNA. So anti-codon's on the tRNA. The codon, of course, is on the mRNA. So depending on what the anti-codon is, depends on what amino acids appear in the amino acid binding site, the amino acid attachment site. So those codons on the mRNA base pair with a specific anticodon on the tRNA, and depending on what that anticodon is what the amino acid that this tRNA brings over. So when mRNA comes out of the nucleus, it needs several things for translation, making proteins. It needs all the amino acids in the cytosol. There's all 20, many of each kind, monomers, amino acids floating around. It needs the small and large ribosomal subunits. It needs tRNA that's hooked up to one of the specific amino acids. And it needs ATP to build the bonds. This is an anabolic process. Okay, it doesn't happen without energy. So the stages. Okay, so we see that the mRNA binds at first when it comes out. It binds to the small ribosomal subunit. The start codon is exposed, and the mRNA slides that way, and when it gets to a certain spot, the large subunit clamps onto it. There's 3 little chambers, cavities, inside the large subunit of the ribosome. And you can see that the tRNA is housed in the middle chamber, the P chamber. Okay, look, AUG is the codon, right? For the start, it's called the start codon, like I said before. If you look at the genetic code, this would be met. The amino acid would be met. Okay, so here's where we left off, right here. Okay, what's going to happen is this codon right here is going to match up with this anticodon, and there's the second amino acid. The first amino acid is always met. Okay, so transfer RNA with the proper anticodon matches the next codon and brings with it the amino acid that's supposed to be the next amino acid in the protein according to that next codon right there. Okay, as soon as that tRNA enters, this bond forms between the two amino acids. And as soon as that bond forms, that takes ATP, by the way. So ATP is used to form that bond. As soon as that bond forms, the mRNA clicks forward. It like a train track, a train car on a train, and what was here shifts to this room, and what was here shifts to the middle room, and this knocks out the empty original tRNA that just lost this amino acid. So he gets out of there. So maybe we should label these 1, then this is 2, 1, 2. I'm labeling the tRNA. Then 1, 2, but then 2 shifts over to the middle position. And this just keeps on going and going until you hit the stop codon. Once you get to the stop codon on the mRNA, remember, mRNA is that strand that's moving through the ribosome. When you get the stop codon, a release factor called a protein comes in. And what that protein does is when it comes in, it breaks this bond right here on the last tRNA, and that whole amino acid chain is released. And then those 2 subunits pop open as well, the 2 ribosome subunits, releasing also the mRNA. Okay, so this is the protein or the polypeptide, and each of these is an amino acid. Depending on what these amino acids, as soon as that protein is released, it starts folding up on itself, giving the tertiary and quaternary and secondary structure, which we talked about before. Okay, again, this is just an example. Here is hemoglobin. Hemoglobin is what makes our red blood cells red. It contains iron right here. The iron molecule is bonded in these groups right here. These called hemes. So these things hold the iron in the hemoglobin. So that this is a quaternary structure because we got 4 strands. You can see red, purple, green, blue. Each of those polypeptides connects to each other, giving this globular structure. So it's just an example of the folding that will happen once the protein chains are formed. So translation does about 20 amino acids per second. So pretty quick. So when you think about it, one of our cells has about 10 million ribosomes. And primary function of a cell is to make proteins, no matter what kind of cell it is. Okay, let's do a little review. What 2 components is a ribosome made of? Well, 2 subunits, but that's not what they're talking about. It's made of ribosomal RNA and proteins complexed together. What is the function of the ribosome? Well, we learned it was the workbench that proteins are snapped together on. It's an enzyme that forms that peptide bond between amino acids. Okay, so there we go, right there. Okay, what is T in transfer? Okay, I just said it. T is transfer. It transfers amino acids to the ribosome. It doesn't. It also refers to the shape, but not really. That's just the way I remember it. It transfers the protein to the DNA. No. Protein doesn't. No. So I say both A and B, but that the T shape is just a memory device. That's not what the T stands for. Shoot, I forgot to push the record button, so let me just talk through this. tRNA has this clover leaf shape. Some call it a T shape. What's causing that shape? Well, it's hydrogen bonding between these parts, between nucleotides, nitrogen bases, in the nucleotide specifically, that causes it to pinch together at these places and then be loose at these places. Okay, so it's because sequence and binding of its amino acids, it doesn't have amino acids. It carries amino acids, but carrying the amino acid doesn't affect its shape at all. The sequence of and binding of nucleotides, that's the right answer, because these nucleotides will base pair, hydrogen bond to those nucleotides on that part of the strand. Okay, how did the anticodon on the tRNA and the codon on the mRNA bind match up? Well, the hydrogen bind is complementary base pairing. So if this was C, we don't have any U's. We don't have any T's, so I can't use T's. CUG. If that was the codon, this codon would be this anticodon would be GAC. These would be hydrogen bonds between them. can't be a strong bond because it's not. You don't want it to stay there. It has to pop off after it does its job. Okay, so what is required to initiate translation? Well, translation is making protein. I need mRNA, ribosomal RNA, and tRNA. I need all three of those. I need amino acids. I don't see amino acids. I have one amino acid here. Of course, that's the amino acid I need because remember, the start codon is AUG. If I looked at my genetic code table, that codes for the amino acid MET. And of course, I need a ribosome to assemble. The two subunits have to clamp together where that tRNA connects to the start codon. So I need all of the above. So be careful about all those above equate questions. What is the first event of translation? Well, the start codon, a tRNA molecule enters the ribosome. Well, ribosome assembles near the start codon. The polypeptide is elongated. No, that's not the first. An uncharged RNA. What do you mean uncharged? You mean a tRNA without an amino acid? That's not true. A ribosome assembles near the start codon of an mRNA. Okay, I agree with that. A tRNA molecule enters the ribosome. No. First mRNA has to be there. The mRNA has to connect to the small subunit first of the ribosome before a tRNA enters the ribosome. As soon as, let's say that's OG, as soon as the start codon goes there, a start tRNA will enter onto it, and then the top clamps down. So it's letter B. If you look back at the pictures, the tRNA went on the messenger RNA, and then that caused the top ribosome unit to clamp down. It's not like the tRNA entered the ribosome. The start tRNA didn't enter the ribosome. The ribosome clamped on after the tRNA was connected to the messenger RNA. It's kind of too picky a question. I would reword this. What is the first step of translation called? Initiation. It's kind of tricky. What does the ribosome do in translation? Well, it's the platform that holds the mRNA, and it provides rooms for the tRNA to link onto the mRNA. I don't see those choices here. It also acts like an enzyme to form the peptide bond between the amino acids of the growing protein chain. So it makes a peptide bond between codons. No, it's not between codons. It's between amino acids in the protein. So let's look at letter B. It makes hydrogen bonds between the codons. There's no hydrogen bonds between the codons. There's covalent bonds between the codons on the mRNA. What does termination and translation occur? Translocation of ribosome. No, that means translocation means shifting positions on the mRNA. That's happening all the time as new amino acids are added. The ribosome gets a stop codon in small and large. Yeah, that's true. RNA polymerase falls off. Wait, RNA polymerase isn't in this picture. RNA polymerase is in transcription. A tRNA brings in an amino acid. No. Remember, on the stop codon, on the mRNA, you don't get an amino acid, you get a termination protein, a terminator protein. Termination factor, as they called it. That's not tRNA. So it's letter B here. What is translation? Duh, you should know by now. It's when mRNA language is turned into protein language. So let's look at these choices. The assembly of the amino acids. Well, really, we're not making the amino acids. The amino acids were already made by another process, chemical process, which we haven't talked about. The amino acids aren't being assembled. The protein is being assembled by the amino acids, so forget this choice. Assembly of polypeptides coded for by the mRNA. Yeah, that's what it is. The making of mRNA. No, that's transcription. Assembly of codons from... Okay, that's transcription 2. What is a gene? It's a piece of DNA that codes for a protein. Or for an mRNA if you want. Or rRNA or tRNA. It codes for RNA. The RNA eventually holds that code for a protein. So, okay, what process relies on complementary base pairing between nucleotides? Well, obviously, when you make DNA, you add new nucleotides to each parent strand. That's base pairing between nucleotides of the new and the old. Transcription, obviously, yes. The nucleotides of RNA base paired to the parent strand, the template strand of the DNA. Translation, yes, you got codons and anticodons base pairing. So it's all those. So a mutation, permanent change, the sequence of nucleotides in a gene, which is in DNA. So this brings about possibly a change in the protein, a significant change in the protein that's encoded by that gene. Okay, so you can have substitution mutations, which means that one nucleotide was mistakenly substituted in for the correct one during DNA replication of the parental strand. So here's the correct DNA codon. I don't want to say codon, but here's the correct RNA, messenger RNA, codon, there would be the protein, the amino acid that would match that. Now, what happened if accidentally you put a thymine was accidentally put in instead of adenine during replication? Well, that's what that's going to do is change the codon. So instead of U, there's going to be an A there. And if you look at your genetic code table, which you'll get on the test, U is a different amino acid than U. So that might not matter too much, but that could be a crucial amino acid that changes. You change that, and it changes the shape of the protein. It doesn't fold correctly, and it loses its function, possibly. So look at this sentence. The sun was hot. Okay, we were going to split that up. Okay, we're doing this because these are like codons. Or you can say they're the sequences on the DNA that code for the codons. Okay, so now, what if by accident you inserted another nucleotide here? It wasn't substitution, it was an insertion. These are called insertion mutations. So what if I put another letter there, like this, A? Okay, so what would happen would be you shift the whole sequence of codons. You'd shift it because you only read in groups of 3. So we would get something like this. At Isu Noa Show T and whatever. It wouldn't even be a complete codon that was coded for by the DNA. Okay, so this doesn't make sense. This is called a missense mutation. Doesn't make sense. So when you have an insertion or a deletion, if you had a deletion, the same thing would happen. So if you had a deletion, you change the color. Let's say it went to T. Okay, I lost the H. So it was Tes Unwa Ash, and then just OT, which wouldn't make sense because something is missing, or it would keep on being messed up down the line on the DNA. So Tes Unwa Ash Ut doesn't make sense either. And that's what they're trying to show here. So you could have a deletion of one nucleotide or insertion of one or two. It can be any number, and you'll get what is called a missense mutation. Now, these are also called frame shift because the whole frame was like your fingers came off the keyboard as you were typing. They shifted, and it makes, have you ever done that? You're typing something on your iPad, and your fingers are off, and you're not looking at the keys, and everything, you think you're typing it right, but it doesn't make any sense because your fingers are off one key position to the right, maybe. So These are called frame shift mutations. So I've just given you a whole bunch of terms here. A deletion is an event that causes a mutation, a deletion mutation. An insertion is an event that causes a mutation called insertion mutation. It produces gobbledygook. It doesn't make sense what the gene no longer has any of the amino acids in the right ones after that insertion or deletion. It's called a missense mutation because it won't form a proper protein at all. It's also called a frame shift mutation because the whole frame has been shifted all the way down the line. And this is again this would be in the new DNA that was produced during replication, DNA replication. Okay, so here's the correct DNA sequence. What happens here, insertion mutation, it looks like they put an A right here. Accidentally, extra nucleotide got inserted. It wasn't substituting anything, it was inserted right there. You can see it. So how does that affect the RNA, the messenger RNA? Okay, it didn't affect that one. This one was changed. This one was changed. This one was changed. So we got everything messed up probably until we get to the end. Now, this is called a nonsense, not missense, but nonsense mutation. When it stops, the protein just doesn't form because you get a stop codon. The protein should be finished. Okay, so this next example, what they do here, looks like they lost, a deletion mutation. They lost, I don't know, one of these A's. I'll say this A. This A was lost. So if you look at, therefore, the codons that we formed, if you scrap out that A, the new codons, the new DNA would be, the mutated DNA would be AAT, TTTC, etc. And the codons would not match what they should be for a proper protein. So UUA and UUU makes a whole different amino acid. AAA and AAG is still the same. Okay, because of redundancy in the genetic code. And then this one's still the same, even though GGG and GGC are different codons, but because of redundancy, they code for the same thing. But depending on what follows, the likelihood of very different amino acids being coded for than the original would happen. So that's a deletion mutation. Both kinds produce missense or missense mutations because the protein doesn't make sense for what you expect it to be. But also, both are called frame shift mutations. Now, some frame shift mutations are nonsense mutations if the protein stops right away or makes a really short protein totally ineffective. And some are missense mutations. So the frame shift mutation causes either a missense mutation or a nonsense mutation. Okay, if you have a substitution mutation and it really doesn't change the type, it doesn't change the type of amino acid that's in the code because the redundancy in the code, you'll get the same exact protein, same exact amino acids. This is called a silent mutation. It has no effect on the produced protein. Okay, redundancy. If you look at your genetic code table, leucine has 6 different codons that codes for it, and so this is called redundancy, and it kind of prevents big changes if there's a mistake. Now, come on, DNA is very careful about proofreading what it does, but like 1 in a 20,000 chance that it'll make a mistake that won't be caught by its proofreading enzymes. So mutations are rare but spontaneous. Now, mutations are not rare if you're exposed to some chemical or radiation energy that cause mutations. So mutagens will increase the rate of mutation. Maybe the DNA proofreading mechanisms won't be able to keep up with all the mutations that occur if you're exposed to these things. So X-rays, gamma rays, UV can cause skin cancer, mutations that causes cell to turn cancerous. So too much radiation can upset the DNA, knock out nucleotides, and so random nucleotides will be deleted in DNA that's already formed. It's already been replicated. So those templates will be messed up. Okay, tobacco, all kinds of drugs can cause mutations. So are mutations always bad? No, they could be silent, and there's no effect to the protein. It could be one amino acid is changed, a substitution, that makes that protein function a little better. So mutations are the engine of evolution. So it's kind of a built-in thing that happens so that if the environment changes and it's not very pleasant to live there anymore, it's hard to live there in that new environment, mutations can give you proteins that allow you to fit that new changed environment and adapt better than the other ones that weren't, with proteins that didn't get mutated. And you'll leave more offspring. And your mutated gene will become the favorable gene in your population. In other words, you will leave more offspring. They'll get that mutated gene. If it was a gene that got mutated in your sex cells, that is, you'll leave behind children that will be better adapted to that changed environment. But that's only if your sex cells get the DNA in your sex cells get mutated, because if you get mutation in a skin cell, that's not going to carry on to your kids, because it's that mutation in the sperm DNA. Okay, changing one nucleotide, making a different nucleotide, or deleting it, or inserting it, can change everything. The primary structure, which is the sequence of amino acids in the protein. The secondary structure, how the protein folds up into alpha helixes or beta sheets. And tertiary structure, or the quaternary structure, it won't fold properly and make the right three-dimensional globular shape. So it's all those. Okay, substitution means one or more of the nucleotides got switched out with something else. So we can tell this one is exactly the same. So there was no mutation there. So that narrows it down to 3. This guy is totally too short. It didn't have substitution. It had a whole bunch. It had 3 that were deleted. And the CCC probably is a substitution, but I just want a substitution only. So let's cross out A. Now, this one could have had a substitution or more, but it's not just a substitution. You have an extra. So that's an insertion. That's an additional. It's not a substitution. A whole new one has been jammed in. So it must be this one, because there's a substitution. G. Yeah, there's only one thing that got substituted, and that would be the A right here with a T. So frame shift would throw this off by 1 letter, maybe 2 insertions or deletions. So this is not even affected, so that's not frame shift. And let's see what that looks like, a substitution. This looks like an insertion that throws everything off. So it has to be this one. A mutation causes DNA is temporary. No, it's always permanent. Always fatal. No, it could be helpful. It could be no effect at all. Permanent. Yes, always permanent. Not always beneficial. All mutations are caused by mutagens. No. Mutations randomly occur. They're necessary to keep a population adapted when the environment changes. We'll learn that in evolution.